All this is Dr. Mobeen Sayed from drbean.com. Welcome to one more show. So, the discussion today is about the mitochondria and a special molecule present in the mitochondria called cardiolipin, and how cardiolipin stabilizes the electron transport chain complexes. This topic is from my book, which I'm writing, and I'm doing drawings. That is why I've been absent from the YouTube lectures for some time. So all the diagrams that you're going to see are from that book as well, and this is the topic from chapter 4. So let's look at it. So these are gifts for humanity, and they are continuing. So today's uh, takeaway is let your mitochondria thank you by protecting their cardiolipins. So in a mitochondria, I think we know that the mitochondria are made up of a double membranous structure. So there is an outer membrane or outer envelope. Imagine that there is an outer bag, and inside of that is an inner double membrane as well. So imagine a bag within a bag. The discussion that we are doing today is about this inner membrane of the mitochondria. On this inner membrane of mitochondria is a very special machine called electron transport chain. It is a complex of 4 units. These are called complex 1, 2, 3, and 4, which are then connected near another molecule which is called ATP synthase. So this whole machine together works to make ATP, which is the energy that our cells use. And anytime that ATP is not produced correctly, or the mitochondria does not function correctly, our cells cannot function correctly, and then whatever is the tissue whose mitochondria is not working correctly will have the tissue related symptoms. For example, if it is brain, then the brain fog would appear. If it is muscles, then tiredness and fatigue would appear, and so on. So today we are talking about this particular complex of lipids called phospholipids called cardiolipins. An important character in this whole structure is reactive oxygen species. Reactive oxygen species are produced when the mitochondrial electron transport chain is functioning. Most of the time, about 2 to 5% of the energy that we have taken in the form of nutrition becomes converted into the reactive oxygen species. These species are actually useful. They are used for signaling, they are used for protecting a cell from harmful pathogens, etc. So it is not that these reactive oxygen species are uh, useless. However, when their quantity increases, when their amount increases, that is when damage starts occurring. Now, the reactive oxygen species are primarily produced in the mitochondria when these complexes, the electron transport chain complexes, are functioning. And when the electron transport chain complex becomes unstable, becomes damaged, then even more reactive oxygen species are produced. Why? Because we are bringing in energy. Let's say we are eating food that is bringing in energy. That energy now needs to be converted to ATP, and ATP is not manufactured correctly because the electron transport chain is not working correctly. The result is that nutrition then starts getting converted to reactive oxygen species, and we end up with a larger amount of reactive species. These then start causing damage. And I've discussed this in the chapter number 4 in my book. So here is the mitochondrial. If we zoom in, this is the outer bag membrane. This is the inner bag membrane. And if you see here, there is this complex or a chain of complexes. This is complex number 1. This is complex number 2. Let me zoom in a little. Complex 1, complex 2, complex 3, complex 4, they are situated next to ATP synthase. And here you are seeing reactive oxygen species that are being produced. So if you see here, complex 1, when it is functioning, it gives off some reactive oxygen species. Similarly, when complex 3 is working, it gives off a reactive oxygen species as well. Those reactive oxygen species can actually turn around and attack the same complexes if their number goes beyond a manageable number. So here, this is the cardiolipins. Now, what are cardiolipins? These green molecules in our membranes. So if I go back for a second to the membrane here, these membranes, so we are specifically talking about the membrane here. Our membrane are called phospholipid bilayers. What that means is that we have a phosphate molecule. With that phosphate molecule, two lipid molecules are attached. Most of the time, one molecule is the saturated lipid or straight, and the other one is unsaturated, or what we say polyunsaturated fatty acids or PUFAs. The other one is usually uh, polyunsaturated, which causes a bend in its structure, which allows various molecules to kind of stay away from each other because of those bends. And that is how the fluidity in the membranes is produced. Now, we are talking about a very special kind of phospholipid. Instead of having one phosphate and two legs of lipid, this one has one phosphate and four lipid legs with it. This is a cardiolipin. This is specialized phospholipid present primarily in mitochondria. Why is it present primarily in mitochondria? Because mitochondria has a need to stabilize and to grasp its electron transport chains. So if you see here, these green characters that are cardiolipins, they are holding the electron transport chain molecules and stabilizing them. And if you look closely, you would see that there are four legs. So this is one phosphate. With that phosphate, there is one leg over here, lipid leg. This is other, another, and another. So with these legs, these cardiolipins grab hold of the electron transport chain complexes. Normally, cardiolipins help make what we say super complexes. A supercomplex is more than one electron transport chain complexes sitting together. For example, 1, 2, 3, 4. This is a supercomplex because individual complexes are complexes, and then many of them sitting together are supercomplexes. Sometimes cardiolipins can hold complex number 1 and two complex number 3s, or sometimes they hold complex 1, complex 3, and complex 4. So they make various clusters of complexes for the electron transport chain to run efficiently and produce ATP. Now, if we are in a situation where we have more reactive oxygen species, and I've done these discussion in the past, for example, um, advanced glycation end products or advanced lipoxidation end products or ales. When they act on the RAGE receptors, they can help produce reactive oxygen species. Carbohydrates help produce reactive oxygen species. Many of the dietary preservatives or colors or dietary impurities can cause reactive oxygen species production. Many bacteria, pathogens, irritants can produce or can cause production of reactive oxygen species. Many substances that can damage our electron transport chain complexes will then cause more reactive oxygen species production. Normally, what happens is electrons kind of flow from each complex to, let's say, from complex 1 to complex 3 or complex 2 to complex 3. From there, electrons are conveyed to complex 4. There they are combined with carbon dioxide and oxygen and water to carbon dioxide and oxygen to make water. And then the energy is used to make ATP. However, the reactive oxygen species that are produced here can then attack the mitochondria itself. So here, if you see, in this diagram, and these are the diagrams from my book. I hope you you like the work I've done on these diagrams. So here what you're seeing is these are the reactive oxygen species that are striking the cardiolipins. And as they strike the cardiolipins, as they hit them and damage them, they cause these cardiolipins to become unstable. Their legs become damaged. And what happens is those cardiolipins cannot hold the supercomplexes together. That cluster starts falling apart. When the cluster starts falling apart, how did it start falling apart? Because the reactive oxygen species attacked the cardiolipins. The result of that is that the electron transport chain becomes unstable. It is not clustered correctly, and more reactive oxygen species start leaking from this damaged complex. Why? Because the electrons cannot transfer correctly from one complex to other. So let's say complex number 1 is not able to send the electrons correctly to electron complex number 3, then the energy will start getting leaked out as electrons, which would then attack the nearby oxygen to make reactive oxygen species that would then attack other things as well, and especially make perinitrite, which are also very damaging for cardiolipin. Similarly, imagine that the complex number 4 isn't working correctly or has become misaligned. And then the complex number 3 would now start trying to send the electron to complex 4, but it cannot. And those electrons would start leaking. And that is another area where reactive oxygen species would leak. So now we have a vicious cycle. On one end, there are more reactive oxygen species that are attacking and damaging the electron transport chain. And then the electron transport chain damage is going to produce more reactive oxygen species. That vicious cycle would then cause not only the mitochondria to be damaged, but also the reactive oxygen species would start leaking out of the mitochondria and start attacking nucleus and Golgi apparatus and endoplasmic reticulum and the enzymes of the system and the cell membranes, the peroxisomes. It would attack whatever comes in its way. Now, there is another very delicate point which I would like, if you would just remember one thing from this whole lecture. When reactive oxygen species attack another protein, a protein, then the reactive oxygen species becomes neutral and the protein is damaged, and that protein may become repaired or recycled. However, when a reactive oxygen species attacks a lipid molecule, for example, cardiolipin, the lipid molecule, when that becomes radicalized, we call it beta peroxidation. When that becomes radicalized, that lipid molecule would attack the next neighboring lipid molecule. And when it would attack the neighboring molecule, it is not that this lipid molecule would now become neutral. Instead, it would stay radical. And now we have two radicals. They both would attack their neighboring molecules and radicalize them as well and damage them as well, which would then stay radicalized. None of them becomes neutral, and they would attack the next neighboring molecules. And that chain reaction continues. This is why lipid peroxidation or lipid damage by reactive species is more harmful, because lipids themselves start attacking other lipids as well. This is the takeaway from this topic. That means protecting your lipids from reactive species is more important than protecting other parts of the cell. Of course, DNA is very sensitive as well, but lipids create this cascade. So then the question becomes, how do we help this situation? I know that most of us know there are various kinds of antioxidants. For example, vitamin C, glutathione, NAC, vitamin E. The important thing, CoQ10, the important thing is to notice that the antioxidants are of two types, actually three types. Water-soluble antioxidants, for example, vitamin C. Vitamin C stays within the water medium of the cell in the cytoplasm. It can neutralize reactive species that are present in the water-soluble environment. Vitamin C cannot neutralize reactive oxygen species or reactive lipid species because vitamin C does not go to lipids. Lipids are fatty structures. Vitamin C does not like fatty structures. It does not go there. So how do we help the fatty structures from getting beta oxidized or radicalized? We use fat-soluble antioxidants. For example, vitamin E or alpha tocopherols. You would see that if you buy omega-3s, you will have alpha tocopherols present in there already, vitamin E. So vitamin E is a very important antioxidant for lipid soluble products. Similarly, CoQ10 is a very important lipid soluble antioxidant, especially for mitochondria. NAC is also lipid soluble. Then there are some that can be lipid and water soluble both. So whenever you are discussing or thinking about getting antioxidants, make sure that you have antioxidants of both type. So now for cardiolipin, the question is there cardiolipin supplementation that can help? And there has not been any decent cardiolipin supplement or drug that can help this situation. So that means the only way to protect our cardiolipins and to protect our mitochondria is to have antioxidants and to keep the inflammation low. Now, I want to discuss one more thing. Once a mitochondrial electron transport chain starts getting damaged because the cardiolipins are damaged, then if the damage is to a specific threshold, after that, this mitochondria cannot recover. Mitochondria have many ways to recover. For example, there can be a damaged mitochondria that can become fused with a healthy mitochondria called mitochondrial fusion and take the enzymatic structures from the healthy one to try to repair its damaged portions. Similarly, mitochondrial fission can occur as well, where mitochondria would separate into two. What happens is one damaged mitochondria might split into two pieces, one piece relatively more damaged, the other piece relatively less damaged. The more damage will become mitophageed, it would become recycled, and the healthy one, relatively more healthy, would start becoming healthier. So that is fission. And then there is a third stage where mitochondria cannot be recovered. And we have to do mitophagy. We have to kill it. We have to eat it up and make new mitochondria. So please realize that it is not necessary that we can just take an antioxidant and the mitochondria would just start healing. Many times mitochondria cannot heal, but we can protect further mitochondrial damage and allow newer mitochondria that would form to work correctly. So some of the supplements that are especially useful for lipid soluble antioxidant function, CoQ10. CoQ10 actually works in the electron transport chain. So if I go back here in the electron transport chain for a second and zoom in, do you see these little structures here? Let me zoom in a little more. Do you see this little structure with two electrons on it? This little robot is CoQ10. CoQ10's function is to transport electrons from one complex to the other complex. There is another little robot you're seeing which has one electron on it. This is cytochrome C. So CoQ10 and cytochrome C are actually responsible for hauling the electrons from one complex to the next one. So CoQ10, you can imagine, is a direct use. It is a useful product for the mitochondria because it works so much within the mitochondria, especially in the electron transport chain. So CoQ10 is a very interesting molecule to keep. Then there is another molecule called MitoQ, which is said to be more affine to mitochondria. It has better affinity to enter a mitochondria and help with the antioxidant behavior and to try to repair the electron transport chain function. Similarly, there is another molecule called SKQ1, which is also a CoQ10 that is affine towards mitochondria better than CoQ10. Then, as I said before, vitamin E or alpha tocopherols, they are lipid soluble antioxidants, must have. And then astaxanthin is another lipid soluble antioxidant. So this is the discussion. Now, if you give me a couple of more minutes, I want to give you a quick preview of the chapter 4. The book actually has been written. I am working on the diagrams. I have reached up to chapter 7 in the diagrams, and I have another. So I've drawn in my absence in last 3 weeks, I've drawn about 120 diagrams, and there are another 150 to go. My hope is that in next 1 month, I'll finish those and then the book would go for publication. So here, this is chapter number 4, mitochondrial meltdown, how ROS turn energy factories into engines of destruction. So that is the complexes I just discussed. So I'm not going to read this, but here is the mitochondria. This is the reactive oxygen species damaging the mitochondrial DNA. Then if you see here, this is the discussion that I just showed you. If I read a little bit of this part, mitochondrial membranes also face significant risk. This is the last part of the chapter 4 of the book. Like the plasma membrane, the inner mitochondrial membrane is rich in lipids that are highly prone to peroxidation. Lipid peroxidation is the radicalization of lipids. This membrane houses the electron transport chain and contains cardiolipin, a phospholipid that stabilizes the electron transport chain complexes. When ROS attacks cardiolipin, the electron transport chain unravels, disrupting electron flow and slowing ATP production. The breakdown of cardiolipin also disrupts the cell's ability to remove damaged mitochondria. Normally, oxidized cardiolipin signals dysfunctional mitochondria for degradation. However, when oxidative stress overwhelms this system, defective mitochondria persist, making the cellular environment even more unstable. And then here is the diagram that I just discussed with you. As mitochondrial function declines, your body struggles to meet its energy demands. This is especially problematic for high energy tissues like the brain, heart, muscles. Neurons rely on mitochondria to produce ATP, which is crucial for maintaining synaptic signaling. Without sufficient energy, cognitive function declines, leading to memory issues and neurodegenerative diseases. Heart cells, which require constant energy to maintain rhythm and force, begin to suffer, increasing the risk of heart failure. Muscle cells weaken, metabolism slows, and fatigue sets in, making physical activity feel exhausting. In response to failing mitochondria, cells interpret the situation as a crisis. They increase glucose uptake and shift towards glycolysis. Glycolysis is a production of ATP without using the mitochondria, but glycolysis is very inefficient and actually toxic, a less efficient form of energy production. But this adaptation comes with its own set of problems. Glycolysis leads to accumulation of lactic acid, which acidifies the cellular environment and worsens metabolic stress. The inability of mitochondria to meet energy demands sets off widespread disruptions in cellular signaling, contributing to insulin resistance, hormonal imbalance, and chronic inflammation. If enough mitochondria fail, cells face a crucial decision: attempt to repair them, eliminate the most degraded ones, or if the damage is too extensive, initiate apoptosis. That is, the cell would decide, hey, I am going to die. Because without mitochondria, a cell cannot really function. Mitochondria provide energy. A programmed cell death. In some cases, apoptosis prevents further damage and contains mitochondrial dysfunction. However, when widespread mitochondrial failures occur across tissues and organs, the cumulative loss of functional cells accelerates the breakdown of biological systems. Mitochondria are not just energy generators, they are gatekeepers of life and death, determining whether a cell thrives, struggles, or self destructs. When oxidative stress hijacks their function, mitochondria become a source of destruction, shifting from supporting life to driving dysfunction. This creates a cellular environment that is starved for energy, overwhelmed by oxidative chaos, and trapped in a cycle of decline, undermining overall health. Yet, even in the face of this relentless cycle, your cells have one last line of defense: autophagy, autophagy, autophagy. This ancient survival mechanism enables cells to clear out damaged mitochondria, remove malfunctioning proteins, and restore order before things spiral too far. When activated properly, autophagy can reset cellular health, slow aging, and even reverse some of the damage caused by oxidative stress. It is your body's final attempt to preserve function and longevity, and it will be explored in the next chapter. So this is the book. This is chapter number 4. If you see here, this is a reactive oxygen species attacking the Golgi complex in a neuron, failing the neuron. Here is the reactive oxygen species, or digestive enzymes that are being formed, which would then digest various things. Here is how insulin is released and the function of the Golgi complexes in that. So this book, I think I have worked on this book with a lot and lot of attention. And as you can see that the diagrams are beautiful as well. Hopefully I'll be done with the diagram soon. Thank you very much for waiting for me while I have been working on the diagrams. I would probably come back again tomorrow and we'll discuss the senescent cells from my book chapter, I believe, 5. And we can have that topic discussed as well. But thank you very much for coming in. Please like, subscribe, and share. If you wanted to support, the links are in the description. And I would see you tomorrow. But do comment, like, subscribe, and share. Bye for now.